Mitosis and the cell cycle. So what's the big deal with mitosis? It's the process in which the nucleus divides to form two new nuclei and two cells with identical DNA. Mitosis is a form of asexual reproduction in simple living organisms. The outcome of each cell cycle is two identical cells. How is it possible that a baby elephant can grow into a very large elephant? Or why do animals shed their skin? All has to do with mitosis. Mitosis begins after fertilization. Remember, fertilization is when an egg and sperm cell combine together, known as a fertilized egg or a zygote. Once the egg has been fertilized, mitosis begins. That one cell is then going to divide to become two, and then it continues dividing, becoming four, and then those cells are going to divide, and this process continues until we have trillions of cells. Why do cells make more cells? There's three reasons why cells are going to reproduce. Remember, this is an asexual reproduction. Asexual reproduction means that we're reproducing by one parent cell. Cells are going to make more cells for growth, repair, and replacement. For the baby elephant to grow into a very large elephant, that's due to mitosis. The cells are making more cells to allow for growth to occur. Repair. If you skin your knee or break a toe, in order for the healing process to occur, your cells go through mitosis. And then replacement. Cells get old and worn out and need to be replaced. They are replaced again through the process of mitosis. Some important terms you got to make sure that you know are, as a review, somatic cells are the body cells. They're produced through mitosis. And we're referring our studies mainly with humans, so humans again have 46 chromosomes, 23 pairs. The homologous chromosomes, each member of a chromosome has a pair. Diploid, remember that was 2 in a total of 46 chromosomes in people. Zygote and somatic cells. And then the haploid are strictly referring to the gametes, only 23 chromosomes, and that's, remember, gametes are just the sperm and egg cells. In looking at the cell cycle, the steps of the cell cycle is G1 phase, that was growth, S phase, which was synthesis, G2, which was growth again, and then the little tiny portion is mitosis. You'll notice that mitosis on the diagram is just a small portion of the cell cycle. And then once mitosis ends, we then go into the final step of the cell cycle, which is cytokinesis. For the portion that's just mitosis, mitosis consists of prophase, metaphase, anaphase, and telophase. And we use the acronym PMAT, prophase, metaphase, anaphase, telophase. So PMAT, and that helps you keep them in order because that's the order that they occur as well. Before mitosis occurs, we have interphase. Interphase remembers the majority of the cell cycle, that's G1, S, and G2. During interphase, chromosomes are copied, the number of chromosomes are doubled, and that has to occur before the cell divides. Chromosomes appear as thread-like coils, kind of like a plate of spaghetti, and which we call chromatin. That's at the beginning of interphase. But during this phase, each chromosome and its copy, meaning the sister chromosomes, are going to change to sister chromatids. By the end, the chromatin is beginning to coil and tighten, and as it coils and tightens, we're going to get the chromosomes that we're familiar with, that X shape. This is just a comparison of a plant and animal cell in interphase. The stages of interphase, you have your growth or G1 phase where we have metabolic activity occurring. Metabolic activity are just functions in order to have the cell continue living. A lot of growth occurs. During synthesis, again, metabolic activity is continuing, but remember during synthesis, the DNA is going to get replicated. The DNA has a copy made. And then G2, which is the growth stage 2, continuing with metabolic activity, but now the cell is going to prepare for cell division. It's getting ready to go into mitosis. We now have prophase, which is the first step of mitosis. Now the cell is going to begin to divide. The centrioles or poles appear and begin to move to opposite ends of the cell. The nuclear envelope is going to disappear. And then we have spindle fibers that are forming between the poles. This is just a comparison of a plant and animal cell in prophase. Metaphase is the second step of mitosis. The chromatids, or pairs of chromosomes, attach to the spindle fibers at the centromere. The chromosomes move to the equator of the cell. You can think metaphase, middle. The equator is kind of the middle of the cell, so when you think of metaphase, you can think of middle. And in our diagram, you can see our little X-shaped chromosomes with the centromeres attaching to those spindle fibers, which are attached to the centrioles. The chromosomes are lined up in the middle. This is showing you a plant and animal cell, both in metaphase, where those chromosomes again are lined up in the middle. Then we go to anaphase, which is the third step of mitosis. The chromatids, or pairs of chromosomes, are going to separate, and they begin to move to opposite ends of the cell. The centromere is broken in half. Looking at a plant and animal cell in anaphase. Telophase is the fourth and final step of mitosis. Two new identical nuclei form. The nuclear envelope is going to reform around each. Chromosomes uncoil and appear as chromatin, threads rather than rods. Back to that plate of spaghetti look. Mitosis has officially ended. Comparing a plant and animal cell in telophase. Cytokinesis occurs after mitosis. It is also the end of the cell cycle. Cell membrane moves inward to create two daughter cells, each with its own nucleus with identical chromosomes. This is a mnemonic device to help you with the cell cycle, interphase, prophase, metaphase, anaphase, telophase, cytokinesis. You can use the IPMAT-C. I prefer meatloaf at tennis camp. Remember also that interphase is going to consist of G1, S, and G2. Then you're going to go into PMAT, which is mitosis, and it ends with cytokinesis. And then just a final diagram, again showing you the cell cycle, starting with interphase. Interphase again is going to include G1, synthesis, and G2. And then you're going to go into mitosis, prophase, metaphase, anaphase, telophase. Mitosis ends and we go into cytokinesis before we go back into interphase again with G1.